great pleasure for me to introduce Dr. Hubert Deluyker as the first speaker. Hubert has been the chief scientist of EFSA, I might say so. You made EFSA what it is today scientifically. At least how it developed and how it will develop. And you trained to become a veterinarian at the University of Ghent. And you also did a PhD at the University of California at Davis. And we had the privilege of going together, struggling to survive in California. And say whatever you want about California, but how to put it, you work a lot. So I would like with these words to introduce and how to translate from this scientific language into regulatory considerations. And EFSA is one of the main translators of knowledge into evidence that can be used for legislation. Hubert, the floor is yours. And you get 25 minutes of fame.
Okay, thank you very much. Well, first of all, thanks to Professor Deluyker for considering EFSA. I think the price to be paid is that after managing to avoid food additives and enzymes for 12 years, now I've got to talk about it for the first time. So we'll see at the end of the presentation whether you guys have invited the wrong person or not. It is indeed a particular privilege, and maybe in just a few words I can say very easily what EFSA is. EFSA is panel experts like Ivar who have served in his capacity as an academician in the Biohaz panel for 12 years. There is no EFSA without Ivars. There also is Leif Busk here today, who as a national representative has made a tremendous impact on us. The foods consumption data collections that we have gradually harmonized throughout Europe wouldn't exist without people like Leif, who gently stimulated his colleagues in other member states to do a little bit like you do in Sweden and other places that have a good tradition of. That's how Europe is built. What also I think as a third person is worth mentioning is your current boss, Per Bregman, who was with EFSA as an employee, just like me, and was the head of the department that actually also houses the panels that do scientific evaluations. And I think Per made the ideal course. He served as an EFSA employee, and now he's back in the Swedish authority. And I think that's the model that I like to see about Europe. I've been based in Parma for over 11 years. We now realize that these agencies are far away from the Brussels decision-making place. So our management board has decided to re-enter Brussels. We have some offices close to the European Parliament. Actually, for the occasion of this meeting, I will show you the new offices. So actually, we're from September 1st. I'm in this building where we have two offices. You know, I'm told it's a very nice neighborhood close to the Parliament. Of course, I'm impressed with our neighbors who have the European Union flag out every morning when I arrive to the office. What more can you wish as the Eurocrat that I am today? Of course, I noticed there's a second flag there as well. So, with regard to the presentation in the few minutes that I've got left, I'd like to introduce you to the EU agency ecosystem. The roots of EFSA have already been discussed a little bit yesterday. I think we need to agree on what the building blocks are of scientific advice. We have a legal framework that's common. We'll get into food additives, food enzymes. Maybe I'll give you a flavor of flavorings, and then some general conclusions. So what is not presented today is a whole series of other things And that's a characteristic of EFSA, that we're faced as an agency with 35 some different regulations. There was a decision made in the founding regulation not to have one regulatory system, and we're facing the consequences of that every day. The EU agency network is one of the well-kept secrets of the EU institutions, and one of the things that Professor Anne Glover did when she was for a short while chief scientist for President Barroso is to go to talk to our ECDC colleagues in Stockholm, and she said, well, why aren't you guys talking amongst each other? And what we're talking about is a network of chief scientists of 13 agencies that give scientific advice. Half of them are in the natural sciences, half are in the human sciences, social sciences. And it's actually astonishing how often we reinvent solutions to the same issues. And in the process, I've been growing my understanding of the social sciences, how they approach scientific evaluation, and certainly my respect has grown enormously for what we think we're under-resourced. I think you're a bit humbled if you see what colleagues that look at fundamental rights, drug addiction, etc. Have to work with. Of course, we have close contacts with ECDC, ECHA, Environmental Agency, and EMA. So after our colleague from ECDC went back to Karolinski, he asked me to chair for a while. I've done that, and now I've turned it over to the colleagues from the Chemical Agency. Just one small point, without getting into politics. There are 13 agencies I mentioned, and the chief scientists of 7 of them are UK nationals. The statement of fact, it's not a statement of opinion.
I'm not saying anything.
The reasons for the creation of EFSA have been mentioned yesterday, and so our remit is indeed scientific advice and communication. I've learned the hard way that if you're not communicating, you're not independent. If your opinions don't go within you know, a reasonable amount of time, 1 to 2 weeks on the web, I don't think you're really independent. So what's our remit? It's been decided, and that's one of the early other decisions that was made, that it's going to be with a focus on food safety. We've had a discussion yesterday of benefits, etc., but that was actually decided, and the name was very much under debate at the time of its creation. It's the whole food chain, which is very helpful, also a challenge, of course, and the environment is involved through our work on plants, feed, animals, also to some extent on food. Efficacy is much less, but I want to come back though for 1 minute, since I won't talk about efficacy anymore, about the famous health claims, because that was mentioned repeatedly yesterday. I'll just, for what it's worth, my colleague from Nestlé can comment on it later. But I remember attending one conference of Food Drink Europe where the CEO of Unilever was asked, well, you know, this is surely a disaster for you as a company. The BBC moderator, of course, had to stimulate the discussion a little bit. And so she said, well, sir, this is clearly a disaster for you, that 80% of the health claims of industry have been turned down. And he said, excuse me, madam, as far as Unilever is concerned, 80% of our health claims have been accepted by EFSA. What we have seen is that those that made frivolous claims have had their dossiers turned down, and we who invest research into substantiating claims, our claims have survived. I think, you know, if you allow me, that's my perspective and my take on it. And I dare to say that we've set a world standard all based on volunteers. So in that sense, I'm still enthusiastic after 12 years of EFSA, but that's only for the experts. So in good spirits, we've been given the name authority right off the bat. I do believe that you are not born an authority like you're like nobility. It's a title that you may acquire as you gain trust. So this whole food chain is handled by 10 different panels. Today we're focusing on the panels that come towards the end. I'm not talking about the NDA panel, which is the one that has handled the health claims. There's one unit that supports it. There's 2 colleagues who promised to bail me out when I really get into trouble with my knowledge about, in particular, enzymes. So that's what's called the FIP unit, and they support those 2 panels. As far as the ingredients is concerned, sometimes I compare it to making a good meal, and I would say that one of the key issues is really the mandate. You can ask a question so that you already direct the answer, and I think we want to be more proactive, more open in the future about any comments that anybody has about a mandate, because the question drives the answer. And then the whole opinion is only as good as the information, the expertise and the methods that you put into it. And on the output side, we have invested quite a bit, and maybe if time allows, I'll get into that. So first of all, on the data. Because EFSA is supposed to be an organization that gives independent advice, where is the data then coming from? Well, that's still defined in the legislation. Of course, the data is going to largely drive what you assess. In fact, the legislation often goes further. It not only defines the areas, but also the quality standards of the studies that are being done, and whether, for example, published studies need to be included, whether they need to be searched systematically or not. We talked about meta-analysis yesterday. Those kind of things are in legislation. They're not necessarily defined by EFSA, although, as mentioned here, EFSA is more often than not consulted before legislative data requirements are issued. If I understand that it's actually the engineers that came up with risk assessment to assess whether planes would crash or bridges would collapse. And if we look at that from that perspective, risk is about the seriousness of an effect, how vulnerable we are to that, and what scale, what groups in the population are affected. The interesting thing, I would say, if you step outside the box, is that in the food safety model we say, well, we don't want any risks. Well, it's very hard to do risk assessment if the only figure you're accepting is 0. So, but this is a dogma which, it's never stated, but it sort of gets slipped into your frame of reference. The other element that is a sort of an assumption is that we look at one single chemical, and then we make predictions about what might happen in terms of risks and how we can avoid those. That's our frame of thinking. I would argue that's maybe not the only way we can think about problems, but that's very much embedded in our processes. So then as EFSA, when we implement this, we may face different situations, and I'll illustrate that. You have the situation where dossiers for new compounds or new uses of existing compounds are submitted by industry. So then the sponsor conducts or sponsors pays for studies which are often GLP. You make an extrapolation of those lab animal studies to humans, and then you also have to compare that with the potential exposure, which is based again on assumptions. What we've made tremendous progress with is, with the last part, thanks to colleagues in the member states, is on the food consumption data. In 2005 we had basically nothing, and we still were doing many risk assessments because our plate was full. Quite different situation is when you're talking about existing compounds, like existing food additives, where there's no sponsor to be found. So then you, as a panel responsible for the same issue, you rely on what's available in the literature. You may have a sponsor that has consistent or conflicting results with what is being published. You may have monitoring programs for the exposure. So there are some strengths, but there's also some weaknesses. And I have observed that as a chemical is on the market for more and more years, there is more and more research, peer-reviewed research. I think it's particularly peer-reviewed research that shows something. If you don't find anything, where are you going to publish it? So on the one hand, you know, industry gets accused of only giving us the results that are favorable. On the other hand, when you go to the peer-reviewed, there is a suspicion that unless things are unsafe, you don't get anybody to publish. So that's part of the fun that we're experiencing every day. The third approach is about import risk assessment. So, you know, in your framework, in your world, you don't look at things that way necessarily, but when I listen to you about the enzymes, we're really talking about things that are changing over time, and that's exactly what we're doing with plant health and animal health. So bugs grow, they are contained, and so the typical and very important work done by our colleagues in the plant health and animal health panels is about risks for entry, risk for establishment, risk for spread. And one of the examples, I would argue, that was mentioned yesterday on this is, of course, the EHEC crisis, which was the busiest period in my whole professional life. It shows, I mention it for 1 minute because it shows what you can do at European level. This bug had been imported by tourists from one area in the world previously, and that's North Africa. And it's thanks to our Finnish colleagues who had a Finnish lady that came back in 2004. You know, you have the local physician, you have the county, you have the national system, and then finally it ends up in an EFSA database. Along with the EU reference lab, who had several cases from Tunisia, you know, we knew it was the only place. And thanks, unfortunately, to the outbreak then that's followed in France, which was indeed apocalypse now, we were able to combine, and unfortunately, I have to admit that all this stuff had come in through my hometown, the port of Antwerp, to be shipped to Germany. I always say it's being cleared by Dutch customs, but that doesn't really help anybody. But the reality is that we import a different ecosystem. This bug was not present in our ecosystem. So imports means you transplant ecosystems, and these seeds are vehicles. It just, you know, happens to be wherever somebody has deposited, without going into further descriptions, this bug on seeds that were drying in the field. And why was it in France an apocalypse now? Because indeed, as mentioned yesterday, this was a project of the kindergarten teacher because they organized Fête du Village, you know, the village little thing. And one of the projects from the kindergarten was to grow those seeds. So the small children, you know, fed it to their parents and grandparents, and several of them were killed. And it was spread all over Europe. So we're in a one way we're lucky that only 50 people died, but it's 50 too many. Getting to your favorite subject, additives, enzymes, and flavorings. One legislation, I'll mention a lot of legislation, just not that I want to stay with it, but it just gives you a flavor of how many different legislations there are. It establishes, this one 2008, and then the implementation in 2011 establishes a common authorization procedure. But for the data requirements, as you see at the bottom, they're different between those three areas. So that's one thing. For new applicants, the system is consistent with what I explained earlier. The dossier goes via the European Commission, who put a letter on top of the dossier and says, EFSA, please assess. And then if we have questions, we go directly to the applicant. So far, so good. As far as additives is concerned, the legislation helps us to get a clear definition. That is, that it's not normally consumed, it's there for technological purposes, and there is some of it left in the final food. On the new additives, more legislation, including definitions and purity criteria. And then when it comes to the dossier, it does mention toxicokinetics, chronic and subchronic and chronic toxicity, genotox, carcinogenicity, repro, and developmental. We'll see to what extent that is implemented then, and how that is implemented rather, in the next slides. So we have a standing working group within the ANS panel that deals with it. For advice on exposure, we have another standing working group. So these are the people that are responsible for the new dossiers. How does it work in practice in terms of this panel? Well, if you, for example, have a 90-day study, if there's a problem there, or if your genotox in vitro results give you a problem, then you go to the next year. So it's not like you necessarily have to do all the studies. It's a tiered approach. And that eventually, as I said, is then matched with the exposure assessment, which is based on assumptions of use. Who knows beforehand how much stuff they're going to sell. But here maybe I can pause for a minute and get to an essential point of the whole issue, and maybe Leif will expand on that. Of course, we start with the hazard identification and the hazard characterization. When we go back to the whole story of glyphosate, which has been hitting the news for many months now, it's been made out to be a big deal, but in essence, IARC was doing a hazard characterization. That is if you give a rat glyphosate, you give it intraperitoneal at a dose that kills 50% of them promptly, do you see any evidence that there might be some indication of carcinogenicity in those cells? And so the answer is yes. In fact, most things in this room are probably, by that definition, cancerogenic. Our question is not whether standing close to a train that passes by at 200 kilometers an hour is dangerous, but what is the risk that somebody jumps in front of it, and therefore do you need to put up barriers? That's risk assessment. The other is hazard characterization, which allows you to introduce the precautionary principle, because you say, well, something might happen one day. So for me, the difference is very clear, and you know that WHO has its panel on pesticides, and they've come to the same conclusion as we have, and we'll see where the EPA stands in a few weeks from now, when they have their hearing. Okay, so the exposure assessment is in one sense very simple. That is that for every category where you're going to use the food additive, you make an assessment as to how much of that is consumed. But of course, the caveat is we're not measuring it in the same groups. We have food consumption databases which have been collected over the years, and then we have assumptions of what concentrations might be present in or will be present once this stuff is on the market. So we're putting A and B together, but they were not measured in the same persons at the same time. Now we're getting into the fun part, which is the re-evaluation. So separate legislation, again, deadlines and priorities have been set in that legislation. And also, to make everything well structured, we give ourselves as EU institutions until December 2020 to go through these assessments in batches. Quickly you see that in fact this whole re-evaluation, as has happened many times in many panels in EFSA, is overwhelming all the rest. So in 13-14 we did 14, in 15 we doubled it, we're going to double it again in 16, and so as compared to the other work it is substantial. What's the other thing that changes dramatically is that, well, all of a sudden sponsors have disappeared here. We have, of course, industry associations, researchers, but there is no dossier here in this whole system to be found. We get one letter from the Commission saying, hey guys, why don't you reassess 300 food additives, and by the way, we'll be really supportive. So we put out calls, and but the results, the responses to the calls are highly variable. The quality of the information is highly variable. It was many of those publications have never been intended to be used in a regulatory dossier, so we're facing data gaps. With a lot of courage, the ANS panel then said, well, okay, we'll develop a conceptual framework, which you can find on our website, and you know, go through a decision tree as to when there is enough information, when there isn't. In this case, we have, as far as standing working groups, because of the workload and also because it makes sense, several working groups. So this is the one on the new applications. This is a whole set of working groups on the re-evaluation. The original legislation says 90 days for reassessment. In this case, we're talking about 1.5 to 2 years, where essentially as EFSA you're sort of putting a dossier together based on the information that you're receiving. So what takes time is public call, you assess the information that you get, and you may have an additional call for data. So we're talking about a different animal. Then also there have been various scenarios developed to try and get more realistic exposures. Especially as our databases expanded, we have more occurrence data coming in from member states. We're able to refine it. Of course, you can always be very conservative, but then it's also the case that you may easily exceed the safe levels that come out of your hazard characterization. How am I doing on time? 4 minutes. Okay, well, I'm halfway through, so the food enzymes. Well, we're very proud to have a definition of what a food enzyme is and a food preparation. In another presentation one day I'll go into that a little deeper. But food enzymes is a completely different beast, because there we are in fact starting de novo. And so we're learning by experience. Not that there wasn't much regulatory and EFSA activity beforehand, but the key date, in my view, is that the legal deadline for submission of those applications, which for which there was no precedent, was March 2015. And we're still interacting and consulting with industry on dietary exposure issues. The European Commission, as I'm sure several of you know, is working on a document, finalizing a document on food processes. I'll skip the data requirements here. I'll also mention that, as you already highlighted yesterday, that there is quite a bit of GMOs in there. So you have a whole GMO requirements coming in potentially as well, which have to do with the organism's parental recipient as well as a donor, and of course the characterization of the modification. The standing working group is again, of course, matching its expertise with the data that come in. This slide has been put in to convince you that EFSA is aware that enzymes are used in the process and are not to be found in the final product. I hope that's convincing enough. And of course that leads us to the challenges of the exposure, where originally a very conservative method, which we will of course probably claim we didn't invent, was put forward. But of course the use of an enzyme is linked to a process rather than a food. So what we're finalizing at the moment is to say, well, let's keep that conservative method. If you can measure the concentration of the enzyme in the final food, well then fine, then we're back to first principles. If not, well, we'll help you to go back from your final food to ingredients and so back calculate. I would say this is not ideal, and I don't have to convince you, because what the concentration is in the final product, if it is present, is of course the relevant issue from a health perspective. So, in other words, it's the concentration in the raw material on the one hand that we have to somehow bring in the same currency as the food consumption information that we have at the end of that process. There was some discussion yesterday about epidemiology, and it's true we're struggling to integrate epidemiology in risk assessment, because as I showed you in the earlier slides, in risk assessment we're looking at one single compound and what are the possible effects. I think, of course I have to, otherwise they kick me out of the fraternity, I have to make a point about epidemiology. Epidemiology, one could argue, it's not completely true, but one could argue that you look at what causes diseases. And then you try to go back and say, well, here are the potential causes or proxies for potential causes. And in one way, if you look at food, you know, we're talking about additives and enzymes and flavorings, but I wonder sometimes if the most dangerous compound that we have in food isn't called calories. You know, so I think from a policy perspective, you can have all these legislations, but at the end of the day, you may also say, well, what are societal needs? What kills people today? And I think, even though being part of this mechanism, one has to always be taking a step back, and there's probably need for people from academia to remind us and the politicians what really are the top priorities for society. It's a bit shaky, but I hope you didn't choose the wrong person after all.